Hey, Vaughn. You're muted.
You can hear me now?
Yeah,
Okay. I say you look good.
thank you.
You ready?
Yes, I'm ready to do it and get it over with and be finished. Not have to worry about it anymore.
I know. So so you were saying, should we two participants, so just you and I. I was wondering whether we could try the what do you call it, breakout room?
Well, you can't, you have to have multiple participants.
Okay. Okay.
And so it's not too hard. I'll
Mhm.
just, like, once everybody leaves
Mhm.
and it's just the 6 of us, I'll just
Mhm.
set it up and send you guys in there.
Okay. Oh, you're recording already? No, what's that?
Yeah, it starts recording as soon as it begins.
Oh, gee. Sorry.
And then, if you end up getting kicked out of the meeting or something, like if the breakout room doesn't work or something, you can just use the code and the password again to log back in.
Okay. Okay.
Good morning.
Good morning.
Morning. Hi, Mike. How are you?
I'm fine. How are you, Yvonne?
Good. They're supposed to be recording, so we don't say anything.
Thank you. You know, you can't count on my good sense.
Yeah, no, I was talking to her and I looked up the recording. I was like, are they recording this? She's like, yeah. I'm like, okay, I won't say anything.
I mean, you can talk if you want to. I'm going to try to chop off the first portion.
You know, it would probably be good to let people know, you know, in addition to me, during the seminar and during the defense afterward, I think it would probably be very good to make sure that everybody knows they're being recorded.
Yeah, of course. I'm going to say that at the beginning.
Okay.
And then the portion after the presentation won't be recorded.
Okay. Whatever works. I never have anything to say that can't be recorded and played back.
Yeah.
One other thing. I've been in a number of pretty big meetings on Zoom lately, and if too many people have their speaker on, background noise accumulates.
Yeah, I think that's what she said. She said people shouldn't have it on, but
yeah, everyone will have their video and audio turned off for the presentation.
Okay, great. How are you feeling? You doing okay?
Yep.
You're doing okay, good.
she's ready to leave my lap. That's the worst thing.
Oh,
that's not true.
Oh.
I know.
You know, and then in a couple of years they're ready to come back.
Exactly. Well, she's next door, so she can come back anytime. Mm-hmm.
I can't do without my morning coffee. And it's only gotten worse since I've been here at home.
Right.
Looks like you're all fueled up, Meredith.
Yes, my mom went and got me Starbucks, so that made the morning a little bit easier.
Oh, yeah.
That's sweet.
Yep.
How many people are there now? Oh, gee, 31 people? Okay, Brett has already taken off himself. Huh?
Yeah, so there's quite a collection of people here.
Okay.
It's good.
Is the your external advisor there?
Yeah.
Okay. Who else? Jessica there? Oh, Jessica there?
Not yet.
Not yet. Okay, I think I see someone. think she's there. Blackburn, UK. Mhm. So when I'm putting myself up, I just say stop video, right?
Say that one more time.
When I put mute and then stop video, no, video will be on you. Oh,
Yeah, so you just want to make sure both the mute button and the
And-
video button have some
Ah,
sort of red X
right.
or red dot on them.
okay. Just making sure I got it right. Wesley just sent me a text that I shouldn't forget. So we think everyone is there?
Yeah, we should be good to go to get started.
Okay. So good morning, everyone, and thank you for joining us today. I'm so proud to introduce Meredith. Meredith has worked in my lab for about 5 years now. When she joined my lab, I had just been in my position for about a few years, but she trusted me enough to join the lab. She was a very young and shy student, and I've seen her grow and blossom into this amazing scientist. I'm so proud of her. When she joined my lab too, she knew exactly what she wanted to do. She wanted to teach in a university. It doesn't matter how much I tried to convince her. She probably had seen me suffer through the grant writing, well, jokes aside. But while she worked in my lab, she took extra hours supervising undergraduates as well as high school students. But she also sought out teaching opportunities at UK, EKU, Transy, Bluegrass Community College. We're extremely proud of her and so happy for her that she recently accepted a tenure track position at Georgetown College starting in the fall. Despite all the other activities, Meredith has been very successful scientifically, experimentally. Within a few years in, she actually had published 2 papers and was co-author for 3 other papers. And I actually used to tease her that she probably needs to graduate right now. And she kept telling me, no, 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 no. But I think she's really matured into an amazing scientist, and we're so proud of her. She's been able to navigate through projects, but I think her talk today would focus on one of the projects which we haven't published on, we're still writing the manuscript on. It's very exciting, it's very novel. She's been able to, I gave her the idea, but she drove the project. Without much ado, I think I would let Meredith now explain her scientific discovery.
Thank you so much for that kind introduction, Yvonne. So before we get started, just so everyone is aware, I'm recording this session today so that people who aren't able to be here can watch it later. And if everyone can just make sure that they keep their mic and their video off, that would be fantastic so that we can try to reduce distractions. But let me go ahead and pull up my PowerPoint here and we will get started. All right, so thank you all for being here this morning. Today I'm going to be discussing some of the work that I've done over the past 5 years to try to elucidate some of the epigenetic mechanisms that are at play in cancer that is promoted by inorganic arsenic exposure. And you may be wondering, why bother studying inorganic arsenic exposure? Is it really that big of a deal or really worth studying at all? And in fact, inorganic arsenic exposure at low doses is a problem not only worldwide, but here in the United States. There are many regions in the United States where people are exposed to inorganic arsenic above the EPA recommended limit, either through lifestyle choices or their occupation, or primarily untreated water. And if we zoom in close to home and look at Kentucky, you'll notice that there's a higher density of people who are exposed to inorganic arsenic above the EPA allowed limit in eastern Kentucky specifically. And this is due in part to the coal mining industry, which is prevalent in this region, and not only exposes people who work in that occupation to low doses of inorganic arsenic, but it also releases inorganic arsenic into the environment and into the groundwater. And many people in this region use untreated drinking water for drinking, bathing, cooking, all of those things. And so they are being exposed to low doses of inorganic arsenic, and possibly over a long period of time if they live there for a long period of time. And this higher exposure to inorganic arsenic we see correlates also to higher cancer rates that we see in this region, specifically lung cancer. Of course, there are multiple factors that contribute to the higher cancer rates that we see in this region. However, it's probable that exposure to inorganic arsenic could be one of the contributing factors. And so that begs the question, how does inorganic arsenic exposure actually contribute to cancer development? And a lot of times, when we think of cancer, we think of metastasis. And metastasis occurs via a specific process that occurs in cancer cells called the epithelial to mesenchymal transition. Epithelial cells, which are found within our bodies lining our cavities and our organs, typically have a cuboidal or a spherical shape. They exhibit polarity with different sides having different characteristics, as well as adhesion to the other cells in the basement membrane. And because of that adhesion, they are stationary and don't move throughout our body. However, in many cases, cancer cells can go through what is called the epithelial to mesenchymal transition, in which they not only lose that polarity and that shape, but they also lose their ability to adhere to the things around them, and thereby are able to develop the property of being mobile and traveling throughout the body. And these types of cells are what we refer to as mesenchymal. They exhibit a more elongated phenotype. They're no longer adhesive to other cells or the basement membrane. And most importantly, they can become mobile. And that is what allows cells to travel to other parts of the body and establish a new tumor, allowing the cancer to become more severe. So how does inorganic arsenic exposure actually contribute to this epithelial to mesenchymal transition process? Well, a lot of times when we think of cancer, we like to think of it in terms of mutations, but inorganic arsenic is unique in that it doesn't actually cause DNA base pair mutations. Instead, it causes what are referred to as epigenetic changes, which are changes to the DNA or proteins that wrap the DNA that don't actually change the sequence of our DNA. But to understand epigenetic changes, I first must introduce you to how the DNA is compacted within our cells. Each cell within our body contains about 6 feet of DNA that must fit within the nucleus. In order to fit within the nucleus, it must be compacted to fit. And so we begin with the double-stranded DNA helix, and first it gets wrapped around a collection of proteins called a histone octamer, forming what is called a beads on a string conformation. Then those beads on a string form a bigger coil to compact further. That coil can form large loops and folds. Those loops and folds can then be condensed into a greater level of coiling until the DNA can become so compact that it can actually form a chromosome during mitosis. However, for the sake of our talk today, we are going to focus on this level of chromatin compaction. So let's look a little bit closer. So like I said, the DNA is wrapped around a collection of proteins called a histone octamer. It's called an octamer because it's composed of 8 proteins, 2 copies of each of the 4 core histone types, H2A, H2B, H3, and H4. And the DNA wraps around this histone octamer about 1.7 times to compact it. And there are several different changes that can occur either to the DNA or to the histone proteins themselves to upset or change this compaction. And so that brings me to the overall hypothesis for my project and the work in my lab, or in Yvonne's lab, in which I'm looking at how inorganic arsenic exposure can lead to epigenetic changes such as DNA methylation and histone variants that I'll introduce you to soon, and how these epigenetic changes not only alter the DNA compaction, but ultimately lead to changes in expression of the DNA and underlying genes that can promote carcinogenesis. So first, let's take a look at DNA methylation. DNA methylation refers to a chemical addition that occurs on cytosine residues within the DNA in which a methyl group gets added. And this DNA methylation can alter the ability of transcription factors and RNA polymerase to bind to alter gene expression patterns. And so I wanted to identify how inorganic arsenic could be altering DNA methylation to possibly promote cancer development. In order to do that, I developed a cellular treatment model in which HeLa cells were treated with inorganic arsenic at 0.5 micromolar, which is an environmentally relevant dose. And these cells were chronically treated for up to 45 days, and I termed these IAS-treated cells, or IAST. However, a subset of these cells were treated with inorganic arsenic for up to 36 days, but then had the inorganic arsenic removed and were grown for an additional 10 days in normal media to determine if changes instigated by inorganic arsenic are permanent or if they can be reversed when exposure to inorganic arsenic is reversed. And I termed this IAS-REV, or IAS-reverse. And then, of course, I had a subset of cells that were not treated with inorganic arsenic. First, in these cells, we confirmed that EMT was occurring by performing Western blot as well as other assays to confirm that known EMT markers were changed in protein levels as we would expect. And we saw the appropriate changes in the IAST cells as we would expect compared to non-treated. And then in the IAS reverse cells, some of these EMT markers reverse back towards a more normal level like slug or encadherin, whereas others such as Claudin-1 or snail maintained their changed protein levels. And so with the cells in these three different treatment groups, I performed a high throughput analysis of DNA methylation changes throughout the genome. And I found that when comparing inorganic arsenic treated cells to non-treated cells, there were over 30,000 individual cytosines that were differentially methylated in response to inorganic arsenic exposure, with about 20,000 of those being hypermethylated or showing an increase in methylation upon treatment, and about 10,000 of those showing hypomethylation or a reduction in DNA methylation at a specific point. However, when I compared the non-treated cells to the IAS-REV cells, we actually see a shift in that proportion with a slightly higher amount of hypermethylation and slightly lower amount of hypomethylation, but also an overall reduction in the total number of differentially methylated spots. So we went from about 30,000 different differentially methylated regions to about 15,000 differentially methylated regions, indicating that some of the methylation changes instigated by inorganic arsenic are more permanent and persist even when inorganic arsenic has been removed from the cells, whereas others are more transient and reverse back to normal when inorganic arsenic is no longer present. However, it's difficult to create a clear picture of what is going on here when you're looking at 30,000 different DNA methylation sites. And so I decided to hone in on several different DNA methylation changes and identify if those DNA methylation changes could be correlated to changes in gene expression. In general, in the promoter of a gene, when methylation is reduced or we have hypomethylation, it increases gene expression. So I looked at several genes that were exhibiting hypomethylation in my results and identified gene expression changes using quantitative real-time PCR. And you'll see that, as we would expect when hypomethylation occurs, an increase in gene expression occurs. I saw that with the genes that I tested, in which gene expression increased upon inorganic arsenic treatment compared to the non-treated. And interestingly, upon reversal, some of these genes didn't go back towards normal treatment level or normal expression levels, whereas PPM1L started to revert back to normal levels, once again suggesting that some of the changes instigated by inorganic arsenic are more permanent while others are more transient. On the other hand, when the promoter of the gene is hypermethylated or methylation increases, it actually decreases gene expression of that gene because the DNA methylation serves as a roadblock or a speed bump to the transcriptional machinery. So I evaluated gene expression of multiple genes that exhibited hypermethylation and saw what I expected in that gene expression was reduced in response to inorganic arsenic treatment. And not only that, but once again, some of these genes indicated permanence in their expression changes or expression trends, while others seem to go back towards normal expression levels upon reversal, thereby suggesting that inorganic arsenic treatment causes some changes in DNA methylation and gene expression that are more permanent and persist, whereas others are more transient when inorganic arsenic is removed. This brings me to my first summary, in which I've shown you that chronic low-dose exposure to inorganic arsenic is a prevalent issue in the United States, and it has been connected to the development of cancer, specifically through the epithelial to mesenchymal transition. This is due in part to epigenetic changes such as DNA methylation, which alters the ability of gene expression to occur, and my studies showed that inorganic arsenic led to many different DNA methylation changes within our cells. And not only that, but they correlated to changes in gene expression, which could ultimately be contributing to cancer development. So this brings me to the next portion of my hypothesis and the next portion of my presentation, which I'll focus a little bit more time on, and looking at histone variants and their role in inorganic arsenic-mediated cancer development. So first, let me introduce you to what histone variants actually are. Like I told you earlier, the histone octamer is composed of 8 proteins, 2 copies of each H2A, H2B, H3, and H4. And there are normal or canonical versions of these proteins that typically get incorporated into the histone octamer or the nucleosome, which refers to the DNA wrapped around the histone octamer. However, our cells also produce variants of these histones that have either amino acid changes or additions or deletions that can be incorporated into the nucleosome at specific places or in specific times to help aid in progressing gene expression changes for specific purposes. And so these histone variants can alter the compaction of the DNA and its accessibility to transcription factors by altering these nucleosome dynamics. Our cells produce a variety of different histone variants, some which have, like I said, large amino acid additions compared to the normal, while others have large amino acid deletions, and some exhibit just small amino acid changes indicated by these black letters or white letters. Several of these histone variants have been identified in being important for promoting active gene expression, and so whenever they are incorporated into a specific location in the genome, they open up that chromatin and allow for gene expression to occur, whereas other histone variants, when they get incorporated into the DNA, actually create a more compact chromatin structure and repress gene expression. Not only do histone variants alter gene expression in general, but they can also serve more specific purposes, such as the histone variants highlighted in purple that are known to be essential for developmental processes and turning on and off specific genes to allow development to occur successfully. Furthermore, some histone variants, such as H2AX, can aid in DNA repair, for example, serving as a marker of DNA double-stranded breaks so that it can recruit all of the appropriate repair machinery. However, just like with anything in our cells, when these histone variants get employed in the wrong place or at the wrong time, they can actually lead to disease states or disease development, specifically cancer development. And recently, histone mutants or variants that have been identified as contributing to cancer development have been termed oncohistones. And so in this example here, you see we have a region of DNA in which a portion is very compact and not open to transcription, whereas a region is more open and is available for transcription. However, when this oncohistone gets employed in the wrong place at the wrong time, it upsets the compaction of the DNA and can alter the gene expression of the underlying genes, ultimately opening up this region of DNA that should be closed and leading to an increase in gene expression, whereas it is closing this region that should be open and preventing gene expression from occurring here. And so the inappropriate alterations in gene expression and in compaction can ultimately promote tumorigenesis. So like I said, oncohistones can be either histone variants or they can also be histone mutants. And recently in the field, a lot of work has gone into identifying many different point amino acid changes that can lead to a histone mutant that has been identified as functioning as an oncohistone in cancer development. So you can see here that there are many different point amino acid changes leading to histone mutants for each of the 4 histone types. However, I found the amino acid changes in histone H2B most interesting because many of these point amino acid changes or single amino acid changes were identified as creating a histone mutant that plays an important role in cancer development. And this was interesting to me because my work actually looks at variants of histone H2B. And the histone H2B variants, unlike some of the variants I introduced to you earlier, do not have large amino acid additions or deletions. Instead, they just have small, spread out point amino acid changes indicated by these letters in black. And so the canonical histone H2B sequences here highlighted in red, and you can see that each of the 10 histone H2B variants just has either 1, 2, or 3 small amino acid changes that are spread throughout the sequence. You may be wondering, could these small point amino acid changes really be having that big of an impact and truly be contributing to cancer development? And that's a great question that I think can be answered and addressed a little bit easier if we look at the structural data for some of these amino acid changes. So first, let's zoom in on amino acid position 39, which in the canonical histone H2B, as well as several of the variants, is a valine, whereas in other variants, it's actually an isoleucine. And you can see from this nucleosome structure that this amino acid position is located at the interface between the histone octamer and the DNA. And if we zoom in a little bit closer, I've superimposed these 2 amino acids over each other. And so the valine in blue, which is the canonical amino acid, is pointing towards that DNA backbone in orange. However, when it's an isoleucine, as it is in several of the variants, it has that extra methyl group and can actually project closer to that DNA backbone and could be upsetting or disrupting DNA compaction around the histone octamer or that interface between the histone and the DNA. Now let's look at a different amino acid position, position 124. And so in the canonical histone, H2B, as well as most of the histone variants, this amino acid is a serine. However, in a couple of the variants, it's actually an alanine instead. And once again, from the zoomed out nucleosome structure, we can see that this amino acid seems to be located at the histone DNA interface. However, when we zoom in, we see that not only is it located at the interface between the histone and the DNA, but it's actually located at an interface between histone H2B in yellow and histone H2A in pink. And this serine forms an electrostatic interaction with an arginine that is present on histone H2A that binds and stabilizes and helps to promote the compaction and stability of the histone octamer. However, when this amino acid is an alanine, as it is in a couple of the variants, that electrostatic interaction is no longer able to occur and could be disrupting the stability of the histone core. And so in seeing that these histone H2B variants could have unique impacts on chromatin compaction and on cancer development, I wanted to understand their role in inorganic arsenic-induced development. However, in recognizing that EMT is not just getting from point A to point B, but is actually a process, I wanted to look at the changes in EMT as a process. And so our lab developed a chronic low-dose inorganic arsenic treatment model in Bs2B cells, which are a lung epithelial cell line, to look at EMT at multiple time points. And so we began with non-treated Bs2B cells, which are in the epithelial state, and treated them with 0.5 micromolar inorganic arsenic for up to 3 weeks, 8 weeks, and 17 weeks, mimicking the early EMT stage, mid-EMT stage, and late EMT stage. Then we actually increased the dosage of inorganic arsenic to 2 micromolar to bring those cells to a fully transformed state up to 24 weeks. And so we brought these cells through the full epithelial to mesenchymal transition from being non-treated to being fully transformed. And you'll see this bar along the bottom of some of my slides to remind you that we're going through the epithelial to mesenchymal transition. So first, I wanted to identify how histone H2B variants could be acting as oncohistones in inorganic arsenic-induced carcinogenesis. In order to do that, I wanted to look at several different factors. First, identifying changes in H2B variant expression, then looking at a potential mechanism of how those expression changes could be instigated, and then identifying where in the genome these histone H2B variants are incorporating. So to begin, I evaluated the expression of the histone H2B variants using quantitative real-time PCR. And so first, I looked at just the expression of the canonical histone H2B and found that it exhibits mild expression changes during IS treatment, with a slight increase in week 3, a slight decrease at week 8, and then a slight increase back up towards normal expression levels through week 17 in 2.0 treatment. However, what's more interesting is when we compare that to the expression changes seen for the histone H2B variants. You'll notice that they exhibit similar expression trends to the canonical, however, their expression changes are more pronounced and more severe. So at week 3, the H2B variants increase, but to a greater extent. At week 8, they decrease in expression, but once again to a greater extent. And then throughout week 17 and 2.0 treatment, some of the histone variants overshoot that normal expression, whereas others don't quite make it back up to normal expression. So once I identified that expression of the histone H2B variants was in fact being dysregulated by inorganic arsenic exposure, I wanted to understand what could be instigating those changes. And in order to do that, I needed to look at a protein called stem loop binding protein. So typically in cells, we think about mRNA having a poly-A tail, and that poly-A tail gets bound and stabilized by several different proteins that allow it to get translated and remain stable within the cell. However, histone mRNA is unique in that instead of having a poly-A tail, it actually has a stem loop structure at the 3' end. And so the stem loop structure, instead of being bound by the typical proteins, gets bound by stem loop binding protein, or SLBP, which is responsible for stabilizing the histone transcripts and allowing them to be translated. However, this story becomes a little bit more complicated when we look at histone variant mRNA, because it not only can have a stem loop structure and be bound by SLBP, but it can also have a poly-A tail. So histone variant mRNA exhibits an either-or situation. In studies done by other labs looking at inorganic arsenic exposure for 24 or 48 hours, it's been shown that acute inorganic arsenic exposure actually leads to a reduction in total SLBP within the cells. And this is due to a reduction in SLBP transcription, as well as an increase in SLBP degradation. And so I wanted to evaluate how SLBP levels are being changed in our chronic treatment model, since it had only been evaluated in a short-term period. So in trying to identify how chronic low-dose inorganic arsenic affects SLBP levels, I once again performed quantitative real-time PCR and found that initially I saw what I would expect, which is a decrease in SLBP upon initial treatment with inorganic arsenic. However, I next saw something quite interesting, in that SLBP levels do not remain reduced. Over time, they actually start to go back up to normal and start to recover and adapt to the inorganic arsenic treatment and return back to normal expression levels. However, once we increase that dosage of inorganic arsenic up to 2 micromolar to bring them to a fully transformed state, SLBP could not maintain its recovery and drop back down to about 50% expression. I also confirmed these expression trends using Western blot of SLBP and saw similar changes in the protein levels of SLBP as I did in its expression levels. And so in my experiments looking at long-term expression or long-term exposure to inorganic arsenic, I saw that SLBP is actually a little bit more complicated than what was thought before, but there seems to be dysregulation in response to inorganic arsenic with an initial decrease, but then a subsequent recovery over time. So other studies have shown that whenever SLBP levels are lowered or are reduced, it thereby reduces the stability of histone mRNA that contains a stem loop, because SLBP is no longer present to bind and stabilize those transcripts. And I hypothesized that because of this reduction in stability, I would see an increase in the polyadenylated version of the histone variant mRNAs in an attempt to compensate for the loss of the stem looped histone variant transcripts. And so I once again wanted to perform quantitative real-time PCR to identify expression changes, but this time I wanted to do it with just transcripts that contained a poly A tail. And so in order to identify if the polyadenylated histone variant transcripts are somehow compensating for a loss of the stem loop transcripts, I first performed poly A mRNA extraction, in which I extracted all of the mRNA from my cells and introduced a magnetic poly T primer, which bound to the poly A tail in any of my polyadenylated transcripts. Then I introduced a magnet to bind and stabilize any of those transcripts that contain a poly A tail, while I could wash away any transcripts that contain a stem loop or did not contain a poly A tail. And now I can perform quantitative real-time PCR with just my transcripts that contain a poly A tail. When I performed that qRT-PCR, I found that the histone H2B variants exhibit similar expression trends, and they all more or less show a reduction in expression in week 3, an increase in expression in week 8, a reduction again in week 17, and an increase in 2.0 treatment. And so they show an undulating pattern with an increase in week 8 and 2.0. But not only that, we'd not only see an increase in the expression of the polyadenylated histone variant transcripts at week 8 and 2.0 treatment, I also found that they created or composed a larger percent or larger ratio or proportion of the total H2B transcripts. And so here I took the polyadenylated transcripts divided by the total and found that, I'll zoom in here on one specifically, that in week 8 and 2.0 treatment, I see a greater proportion of the total H2B transcripts being polyadenylated. And so we not only see an increase in expression of those polyadenylated transcripts, but they're actually creating a greater proportion of the total transcripts. And so this undulating pattern at first didn't make a ton of sense until I compared it to the changes in expression that we saw for SLBP. So here you'll see the expression trends for SLBP in blue, and then the general expression trend of the polyadenylated histone H2B variants in pink. And so what I found was that initially we saw that drop in SLBP, and like I hypothesized, if there's a reduction in SLBP, there would thereby be a reduction in the stability of the stem-looped H2B mRNAs. And I hypothesized that because of that, the polyadenylated histone H2B variant transcripts could be compensating for this loss of canonical histone transcripts. And that's exactly what I saw. So upon the reduction in SLBP levels, there was a subsequent increase in the expression of polyadenylated histone H2B variants. However, once SLBP levels begin to recover and go back up towards normal, we no longer have that need for compensation, and so expression of the polyadenylated H2B variants goes back down. However, once we brought the cells to full transformation and SLBP levels were reduced again, we once again get a subsequent increase in the polyadenylated expression of the histone H2B variants. In an effort to complete this story, I also wanted to identify the stability changes of the histone H2B variant transcripts as well as the canonical H2B transcript in response to inorganic arsenic. And while these are preliminary results, I found that in measuring the half-life of the histone transcripts in response to transcriptional blocking, I found that the stability of the histone variants actually begins to be greater than the stability of the canonical histone transcript over time in inorganic arsenic treatment. This brings me to my second summary, in which I've shown you that histone mRNA canonical has a 3' stem loop structure that gets bound and stabilized by SLBP. However, histone variant mRNA can have either a stem loop structure or a poly A tail. In response to chronic IS exposure, SLBP levels are dysregulated, but whenever SLBP levels are lowered or reduced, we see a reduction in stability of stem loop transcripts with a compensation and upregulation of polyadenylated histone H2B variant. So this suggested to me that histone H2B variant transcripts are upregulated and polyadenylated to a greater extent in response to inorganic arsenic treatment, and thereby could be incorporating into the genome more than normal or in the wrong place at the wrong time, thereby serving as oncohistones. And so I've shown you how inorganic arsenic altered H2B variant expression, as well as the mechanism of SLBP that could be leading to those H2B variant expression changes. And so the next logical point for my studies would be to try to identify where these H2B variants are being incorporated into the genome so that I could identify specific genomic regions or specific genes that are being dysregulated by the misincorporation of H2B variants to ultimately contribute to cancer development. However, determining the genomic location of the H2B variants requires the use of ChIP-seq or chromatin immunoprecipitation, which requires the use of very specific and very effective antibodies. And like I showed you earlier, these histone H2B variants only have very small amino acid changes that are spread throughout the amino acid sequence, making it very, very difficult to create specific and effective antibodies. Throughout my time at UK, I have been in collaboration with Thermo Fisher to test a variety of different antibodies to the histone H2B variants. However, you'll notice that they don't really exhibit much specificity to any of the histone H2B variants that I tested them against. The only two that showed a little bit of promise were 48430, which shows a little bit of specificity to variants J and K over the others, as well as 48815, which shows a little bit more specificity to B compared to the others. However, these antibodies are not strong enough to be able to perform chromatin immunoprecipitation, and so I had to shift the focus of my experiments. So instead of trying to identify where in the genome the histone H2B variants are being incorporated, I instead decided to look at how H2B variants influence histone octamer and nucleosome dynamics in general, due to these structural changes that seem to be present that I introduced to you earlier, in that the histone H2B variants can lead to changes at the histone DNA interface, as well as histone-histone interfaces. And so I also was interested in looking at how inorganic arsenic contributes to changes in octamer and nucleosome compaction, because my studies indicated that chronic inorganic arsenic treatment actually leads to global chromatin compaction using the salt fractionation assay. And so in this assay, I lysed cells to extract the chromatin and digested the chromatin into small fragments. Then I incubated those fragments in increasing concentrations of salt from 80 to 150 to 600 millimolar. And the less compact that the chromatin is, the less salt it will take to elute that chromatin. And so less compact chromatin will elute in this fraction, whereas the most compact chromatin will elute in this fraction. And so I found that throughout inorganic arsenic treatment, there seems to be a shift from pretty moderate chromatin compaction to a greater extent of chromatin compaction throughout chronic inorganic arsenic treatment. Not only that, but using the two antibodies that seem to exhibit a little bit of specificity from Thermo Fisher, it seems that there are histone variants present in the most compact chromatin fraction. And so it's possible that these histone H2B variants that are being dysregulated in response to inorganic arsenic exposure could be contributing to this greater chromatin compaction that we see. And so in order to identify how the histone H2B variants are influencing octamer and nucleosome stability and compaction and dynamics, I wanted to look at a couple different facets. First, looking at octamer stability, then nucleosome stability, and finally, how H2B variants influence DNA compaction. In order to do these studies, I have to make the nucleosomes, which is not a trivial process, and this begins by expressing and purifying each of the individual histones, as well as each of the variants that I would like to study of histone H2B. I have to use dialysis to create the histone octamer itself, and then go through several more rounds of dialysis, as well as sucrose density gradient and purification by FPLC to create the nucleosome in which 147 base pairs of DNA is wrapped around the histone octamer for these studies. And since this is an arduous process and takes a decent amount of time and allows for low yield of the final product after all of these steps, I decided to focus my studies on 4 different histone H2B variants instead of all 10 of them. And so, of course, I had histone H2BC, or the canonical, in my studies, as well as B, D, J, and K. And I chose these for a couple different reasons. First, they exhibit all of the different 4 possible combinations that you can get for the amino acid position 39 and 124, which I showed you earlier in the structural data. Furthermore, the antibodies that we are collaborating with Thermo Fisher for seem to exhibit specificity to J, K, and B. So if these antibodies become more successful through refinement in the future, we could potentially study B, J, and K using antibody-based techniques. And so first, I wanted to look at how histone H2B variants alter octamer stability using the thermal stability assay. In this experiment, I begin by having my protein of interest, which in our case is a histone octamer, in the presence of a Cipro orange dye and slowly increasing the temperature. As the temperature increases, the protein will eventually begin to denature, and Cipro orange will bind to the denatured protein and begin to fluoresce, which can be detected by an instrument to form this melting curve here, in which we can mathematically determine the melting temperature of the histone octamer. And the higher temperature that the histone octamer melts, that suggests the more stable it is. And so not only did I look at temperature-based stability, but histone octamers and nucleosomes are also sensitive to changes in salt concentration, with higher salt concentrations actually leading to greater destability of the nucleosome. And so I performed these thermal stability assays at several different salt concentrations to ensure that the trends in stability that I saw were not just due to a specific salt concentration, but were due in fact to the stability of the histone octamer. So I had 5 different histone octamers containing either canonical H2B or H2B variant B, D, J, or K. And this is a representative curve from one of the salt concentrations. And you'll see initially that it seems like the histone octamer containing canonical histone H2B seems to melt at a lower temperature than the variant containing octamers. And that is in fact what I saw across all salt concentrations. And here I have each of the salt concentrations that I measured across the x-axis, and then the determined and calculated melting temperature along the y-axis. You'll notice that there isn't any noticeable difference in the melting temperature of the octamers containing the histone variants. However, there is a very noticeable difference in that at each salt concentration, the canonical histone octamer melts at a lower temperature than any of the octamers containing the histone H2B variant, suggesting that the canonical histone octamer is actually less stable than any of the H2B variant containing octamers. Next, I wanted to perform the same experiment, but this time with the nucleosome. So having 147 base pairs of DNA wrapped around the histone octamer to determine if the presence of DNA alters those trends in stability that we saw with the octamer. And you'll notice here that for most of these, they exhibit 2-step melting, in which there are 2 different melting curves. And this mimics or indicates that we're getting 2-step dissociation, in which the H2A, H2B dimer leaves the nucleosome first, and then the H3, H4 tetramer leaves next. And since we're focused on the influence of the histone H2B variants, we'll focus here on this first melting curve. And so once again, I tested across multiple salt concentrations, and this time I saw a slightly more complex trend in the melting temperatures of our nucleosomes containing the different H2B proteins. So interestingly, histone H2BD melted at a higher temperature than the canonical histone H2B nucleosome in red, whereas the nucleosomes containing the histone variants J, K, and B actually melted at a lower temperature overall compared to the canonical nucleosome. And this is interesting because D, which melted at a higher temperature, does not exhibit either of the amino acid trends seen at position 39 or 124, whereas the variants that do exhibit amino acid changes in these 2 positions melted at a lower temperature than the canonical, indicating that when the DNA is involved, the specific amino acid changes that are present on these variants play a more specific role in altering and modulating the stability of the nucleosome. Next, I performed an experiment called the MNase digestion assay to identify how histone H2B variants alter DNA compaction and accessibility. And so in this case, I had nucleosomes either containing the wild type or canonical histone H2B or one of the histone H2B variants. And then I introduced MNase, which actually chews up the DNA to digest the DNA and over several different periods of time. And so I was able to identify different banding patterns or the ability of MNase to digest the DNA when the canonical H2B was involved or one of the variants. And in the results, you'll see that the more banding that occurs on the gel when I run these different timed products on the gel indicates greater digestion because there are more subnucleosomal DNA fragments. Whereas if the DNA did not get digested by MNase as much, you'll see a reduction in the banding patterns. And so in comparing the nucleosome containing H2BC to those containing the histone H2B variants, it seems that the canonical nucleosomal DNA is actually more susceptible to digestion because we saw an introduction of those subnucleosomal bands at an earlier time point and a reduction in the full DNA fragment much faster compared to any of the DNA found in the nucleosomes containing the H2B variants, which we also measured here on this graph by measuring the disappearance of that top band. And so through these studies, I identified that alterations to nucleosomal stability and compaction are very uniquely influenced by the different amino acid changes that are present in each of the histone H2B variants. So this brings me to my third summary, in which I've shown you that chronic low-dose exposure to inorganic arsenic leads to the epithelial to mesenchymal transition that allows cancers to metastasize. Part of the progression of this EMT process is due to dysregulation of SLBP levels, which leads to dysregulation of histone H2B variant polyadenylated transcripts, primarily an upregulation of the polyadenylated transcripts whenever SLBP is reduced. If these histone H2B variants are being incorporated into the nucleosome in the wrong place at the wrong time, they're actually altering the stability of the nucleosome and the chromatin compaction because of their misincorporation. And because of their misincorporation into the genome, they could be dysregulating gene expression patterns that are ultimately promoting carcinogenesis. This brings me to my conclusion, in which I've shown you that chronic low-dose exposure to inorganic arsenic is a prevalent and important concern within the United States and globally, and it leads to cancer development in part by promoting the epithelial to mesenchymal transition. In an effort to fill in the gap here, I have identified how epigenetic changes such as DNA methylation can lead to gene expression changes that promote carcinogenesis, as well as histone variants, which can also alter chromatin compaction and gene expression trends to promote carcinogenesis. So for future directions, as this project continues in the lab, there are more experiments planned to further measure other properties of nucleosomal dynamics, such as nucleosomal sliding, in which the DNA can rotate around the histone octamer, remodeling, in which the histone octamers are removed or replaced, as well as DNA accessibility with a more powerful FRET system. Furthermore, the lab is planning to, and is collaborating to produce tagged histone H2B variants to introduce them into these 2B cells to help us determine the genomic location of these histone H2B variants, since antibody-based techniques are not available. And finally, on a grander scale, I look forward to seeing how this research progresses in the future to identify how multiple epigenetic marks can actually work together to carry out a specific purpose. While I only looked at DNA methylation and histone variants in isolation, it's probable that many different epigenetic marks are working together and exhibiting interplay to carry out specific functions and carry out specific changes to chromatin compaction and gene expression within the cells. So this brings me to my acknowledgements. I, of course, want to thank the Von Dufay-Mittendorf Lab, both past and present members who have helped me along my journey here at UK to develop as a scientist and to do my work and have an exciting project to present for you today. I specifically want to thank Yvonne for her guidance and mentorship and friendship throughout the past 5 years. She took me as a pretty inexperienced scientist and helped me to develop the skills and abilities that I needed to be successful in graduate school. I also want to thank a few specific people who have contributed to this work. Wesley not only is my lab partner and will be taking over this project once I leave UK, but has also become an incredible friend and someone that I lean on a lot at work and in life to help me maintain a positive attitude and celebrate when things are going well and look at the bright side when things aren't going well. I also want to thank Abby Pfister, Rama Nasri, Kelsey Arthur, and Rayhan Ganta, who are students that I've had the pleasure of training and working with in the lab over the past 5 years. I'm so thankful for their friendship and their contributions to the projects and for their trust in me to train them as scientists. Beyond my lab itself, I want to thank my dissertation committee, including Dr. Brett Spear, Jessica Blackburn, and Mike Freed for their support and input and encouragement throughout the past 5 years as my dissertation committee, as well as Dr. Stephen Van Lannen for serving as my outside examiner today and helping me to refine this project in the final stages. I want to thank the biochemistry department, faculty, staff, and students for all of their support throughout the past 5 years. It's been a wonderful place to work, and I'm so thankful for all of the people who have played such an important role in my career, as well as the funding sources of the NSF and NIH for allowing the financial support for this work to be done. Beyond the people at UK that I am thankful for, I also want to thank my mentor from Moorhead State University, Dr. David Payton. He's been a really important friend to me and someone that I look up to, and he's the reason that I want to become a college professor, and he's the reason that I am getting to live that dream out in the fall. I'm so thankful for his friendship and the way that he has guided me over the past 9 years as a student and colleague and friend, and I could not have made it to this point without his support and encouragement and guidance. I also want to thank all of my incredible friends who have walked with me through this journey. I have not done it alone, and I couldn't have done it without the support of many, many people. Specifically, I want to thank Hannah, Jessica, and Sarah, who have been with me since my freshman year of college and have walked with me through a lot of different life stages, and I'm just so thankful for their friendship and so excited that we get to be in each other's lives. And finally, I want to end by thanking my family. Everyone says this, but I think I have the most fantastic family in the world and the best parents, the best sister, and they make my life a lot of fun, but they also make sure that they encourage me and help me achieve my dreams and have always supported me in everything that I've done, and I'm so thankful for them. So that concludes my presentation, and I can now take any questions.
So if you're going to ask a question, can you unmute and then we can go from there.
This is Stephen Van Lannen. I can ask a question since no one else is asking you.
Okay.
I have a question just about the second part of your project, in that you were attributing the melting temperature changes to the DNA compaction of these variants to the point mutations in the sequence. My question is that I know that histones are often post-translationally modified themselves, such as methylation and acetylation. Do you think that would be worthwhile to look and see if there are changes in those histone variants of that? Not necessarily that, so maybe your changes could be actually a function of the post-translational modification of the histones and not these simple point mutations, which I find surprising because they're pretty subtle in terms of the changes.
Absolutely. So since the histone octamers and the nucleosomes were made from recombinant proteins and were not produced in cells, for those specific experiments, I was able to ensure that there were no other post-translational modifications or anything like that. But in talking about the interplay, I can't wait to see how histone post-translational modifications work in concert with histone variants and the different amino acid changes, and as well as DNA methylation, because I'm sure that all of those things are working together. They're not just working in isolation, and I can't wait for future experiments in the field to do exactly what you're saying, of looking at those changes together to see how histone post-translational modifications, as well as the amino acid changes seen on histone variants, could be working together.
Thank you.
Thanks.
Hi, this is Jeremy Wood. Histones also have roles outside of the nucleus and outside of the cell, such as in neutrophil extracellular traps, and free histones can serve as damage markers. Have you looked at whether these histone variants affect the functions of histones outside of the nucleus?
That's a great question. The explanations that I gave for the different histone variants and their functions is about as far as the field has come in identifying specific functions and roles of these histone variants. Because there are so many of them, there's still a lot of work to be done to identify the functions outside of just chromatin compaction for each of the variants. But I imagine that the variants probably do play an important role in other situations and in other contexts besides just chromatin compaction.
Thanks.
So this is Michael Freed. I had a kind of a general question to put things in context.
Okay.
You have been talking about inorganic arsenic species. There are a lot of them.
Yes.
Which are the most prevalent in terms of our local environment, and which are the most bioavailable?
That's a great question. So sodium arsenite, which is the 3+ form, is the most detrimental because it has that lone electron pair. And when looking at inorganic versus organic, the inorganic species are much more dangerous than the organic forms.
So, but danger can be measured a lot of different ways. It can be measured in terms of toxicity to an individual organism, or it can be measured on an epidemiological basis. So, you know, in terms of a population, you may have something that's relatively non-toxic that has a much bigger detrimental effect on human health than something that's much more toxic.
Mm-hmm. Yeah, several different types of arsenic species are identified in different regions, so it's not necessarily just one arsenic. But sodium arsenite specifically has been most focused on in the field as being the most carcinogenic and one that people are exposed to in the environment.
Any more questions?
All right, well, thank you all so much for joining me this morning. If you wouldn't care to go ahead and log out and leave the meeting, unless you're in my committee. Once again, thank you so much for joining us this morning and for listening to my presentation.
Hey.